Okay, good afternoon, everyone. And this afternoon, we're going to start our discussion about one of the most important topics in forensic science nowadays, and that is the use of DNA as a forensic tool. And before I start, let me just get some housekeeping done. Please have a look. I've updated the calendar just for our last few classes. We'll try and stick to that. The most important change to note is that your final exam, will be available for the whole of that weekend, for the Saturday and the Sunday. And it will be available until Sunday afternoon at 4 o'clock, not as usual. We usually have it till Sunday evening at 11, but I have to cut it off on Sunday afternoon because I need to start getting grade sheets done. Please note also that the due date for your final papers, which is our last Thursday class day, by 4 o'clock, please, those papers must be submitted to me. I have to grade them and get them into the grade sheet. There is something a little bit strange you'll notice about the calendar, and that is that our very final class, which is the Monday, is obviously the day after your final exam. That is because I have a movie for you to see there. Obviously, it's not examinable. And it becomes voluntary for you, but a very important movie, which I want to finish off the class with. And I do want you to see it, whether it's examinable or not. It's important for you, and I ask you to please indulge me and watch that movie. Okay, so today we're going to start our discussion about DNA, but I'm actually not going to talk for very long, because there are two movies which I want you to watch today in lieu of class. I'm just going to introduce the topic to you, and then you can move on to watching those two movies. Please do watch them. They're very important, and they are examinable. Your next test is this weekend, and that includes today's material. Okay, so DNA, yes, it's become an almost indispensable forensic science tool where DNA is available from a crime scene. It's very important to realize that DNA is not always left at a crime scene. It frequently is, but not always. And you can't use DNA if there's no DNA to use. Again, what we're doing is we're taking a question sample and we are matching it to a standard. We have to link our DNA sample to someone if it is to be useful. The reason that DNA is becoming so indispensable is that where an identification can be made, where a DNA sample can be linked to a person, that linkage can be done with great fidelity and with statistical certainty. We can state very precisely what the chances are that somebody else would have that particular DNA and share that particular DNA. And the chances are usually extremely remote. So DNA identification has a very, very high degree of validity, and it presents very powerful evidence. And you will realize that it has been used both to convict and to exonerate. There have been many cases where wrongfully convicted people have been exonerated by DNA evidence. It's extremely important for us to understand something about DNA. So just to start off the discussion, I'm just going to tell you a little bit about DNA and how it's made and everything. This will be amplified in the movies that you watch, and then later on as we continue our discussion. You probably have heard before that DNA stands for deoxyribonucleic acid. It's a long name for a very, very, very long molecule. These are huge molecules. And we're going to see that there are several of these molecules in every cell of the human body. all of our cells, all the cells of the body, contain exactly the same DNA. It doesn't matter whether you're talking about a liver cell or a brain cell or whatever. If you look inside the nucleus of the cell, you will find exactly the same DNA in each. It's just the different parts of DNA function in different cells to give them their individual character. DNA is the hereditary material. It is a polymer. That is, it's a very long biological molecule which is made up of individual parts joined together. And in actual fact, the basic structure of DNA is extremely simple because it consists of their making up this polymer, these very, very long molecules. We only use 4 individual units, and those are the individual nucleotides. So nucleotides are joined together to form the long molecule of DNA. And there are only 4 of those nucleotides. And now you need to remember these. They are adenine, guanine, cytosine, and thymine in DNA. Adenine, guanine, cytosine, and thymine, often abbreviated to A, G, C, and T, but you must know the names that are associated with them. Now, those bases, those nucleotides rather, can occur in any sequence. You can have AAA, G, C, A, T, T, A, T, etc., etc. But they, in the DNA, they occur in a specific sequence. Actually, they occur in units of 3 bases. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Each of those little sequences, which is called the DNA triplet, encodes for 1 amino acid. And amino acids are the subunits of proteins. So the DNA bases are in a sequence. And those sequences code for proteins. And you can basically assume that we have 1 recipe for 1 protein. And that recipe we call the gene. That is what a gene is. A gene is the sequence for a protein. It's the DNA sequence which codes for a specific protein. So all of our DNA in our cells is organized, and it's organized into genes. And we inherit those genes from mother and from father, both. In fact, we inherit 1 copy of a gene from our mother and another copy of the same gene from our father. They may not be identical. You may inherit a hair color gene, let's just say a hair color gene. You may inherit a hair color gene, blonde from your mother and brown from your father. So the genes code for the same thing, hair color, but they are slightly different. They may be slightly different. So you inherit 1 copy from mother, 1 copy from the father. And in our cells, the DNA is organized into separate strands of DNA, which comprise the chromosomes. And we, in human beings, we have 46 chromosomes in total. We have 23 chromosomes from the mother. We have 23 chromosomes which we inherit from the father. But because of the processes that take place during sexual reproduction, there's all sorts of things happen. Genes get shuffled and everything. We end up, in the end, unless you are an identical twin or you have an identical sibling like a triplet or whatever, every genome, which is the sum total of all the hereditary information, is unique. Your genome, unless you have an identical sibling, your genome is entirely unique. And we can calculate the chances that somebody on the other side of the world, by pure chance, would have the same genome as you. And it turns out that when we work that out statistically, it is 1 in far more than human beings have ever existed on the planet. That is the chance that somebody else will have the same genome as you. It is as remote as stars in the Milky Way. And for this reason, DNA has extremely powerful individual character. The DNA, sum total of DNA, the genome in total is enormous, absolutely enormous. And the chances that that is going to be reproduced in somebody else is vanishingly remote, unless you're have an identical sibling, in which case it is the same. So that is the background. That is the background to why DNA is so important. Now, DNA consists of actually of two molecules. The whole DNA molecule has two halves to it. And it describes a double helix in space. So here's the one molecule, here's this blue ribbon here. That's one side of the molecule there. And here is the other side of the molecule here. And this backbone here is very, very strong. This is extremely strong backbone. These are covalently bonded parts of the nucleotide which form this backbone. It's actually each nucleotide has a phosphate and a sugar attached to it, and those get joined together to form the backbones here. Then across the middle here are the parts of the nucleotide which actually give each one their character. So there are four. Remember, there are four kinds of nucleotides, and here they are. I just need to try and move something out of the way. Here they are here. Here's guanine here. Here is cytosine here, thymine here, and adenine here. And you'll see that they link across the middle. They join across the middle, and they join these two halves of the molecule together into this long double helix in space. There is a very strict rule in construction of DNA, and that is these matching bases across here. Guanine always matches to cytosine. Thymine always matches to adenine. It doesn't matter what order they come in here, they will always have a matching order on the other side. So have a look here and you'll see this sequence here on this side of the molecule is G, T, C, A. The matching molecule on the other side has to be C, A, G, T, because G matches to C, C matches to G, T matches to A matches to T. This can be in any order. The order of the sequence of nucleotides is what determines what the gene does. The sequence of nucleotides determines what protein that gene codes for. And it is matched on the other side by matching bases. So have a look here. Here it is again. Here we can see the actual structure of the DNA itself. It consists of a sugar there and a phosphate group there. And here's the base here. So this is adenine here. Here is the sugar. Here is the phosphate. The only place that these can join together is through this. They join phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That is very, very strong. DNA is a very strong molecule because of this, and it's a very stable molecule. Here's the other side here. The sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And here are the matching bases. If this sequence here is A, C, G, T, this sequence on the other side has to be A, C, G, T, because T matches to A, G matches to C, G matches to C, T matches to A. This is very important because it gives DNA one of its most important characteristics. And that is that every time a cell divides for whatever purpose, whether it's in growth, whether the cell is dividing to replace itself, or whether we're dividing cells to start to produce gametes, eggs and sperm, the DNA has got to replicate itself. It has to double up. And in order to double up, in order to replicate DNA in the cell quickly and efficiently, is very easily done, because all we have to do is separate these strands one from the other and provide the nucleotides that match. And they can be fitted in. It's done enzymatically in a very controlled fashion. But what happens is that we're going to split the DNA molecule in half, like this. And we're going to use this sequence here to recreate this strand there. And we're going to use this sequence here to recreate this strand here. And when we've finished, we'll end up with two strands which are identical to one another. And that is all because A matches to T and G always matches to C. Doesn't matter what their order is on this strand, the matching strand must fit with them. Okay, so you can do it yourself. Have a look here. And what do we have here? A strand, one DNA strand, and it reads AGT, AA, GC, TG. Now remembering that A always matches with T, T always matches with A, G always matches with C, and C always matches with G. Recreate this strand here. And what do we have? We have A matches to T. So we start T, C, A, T, T, C, G, A, C. Okay, so what you've done is you've doubled that up. You've recreated, you've created the matching strand there. Have a look at this second example, T, C, A, T, T, C, G, A, C. Same story, A, G, T, A, G, C, T, G. And I've tricked you, because in fact these match to one another. What you've done is you've split a DNA molecule apart and you have just replicated it. You've just doubled it up there, exactly like the cell does. That's exactly what the cell does. It does it with enzymes that carefully shepherd in the correct things, but it always matches A to T, C to G, and the cell can very quickly and easily replicate itself, replicate all of its DNA when it needs to do so. Okay, so with that very brief introduction to DNA, I want you please to watch these two movies. And please be careful, these are examinable, okay, in your test coming up on the weekend. So I will leave you with that, and you can fill in the rest of the time for this class by watching these movies, neither of them particularly long.